Taran Kermani, and my research is investigating the function of GT1 and GT2, two genes, in the onset of dark-induced senescence. So, senescence is a process during which wide-scale metabolic, physiological, and structural changes occur in plants in response to developmental cues and environmental stimuli. In terms of my research, the process of senescence can be thought of as the process of aging. So, as plants senesce, they age. Previous research in the lab identified a mutant Arabidopsis line in a gene called COG7 that has an earlier onset of senescence than wild-type plants, specifically when kept under prolonged periods of darkness, which for sure I will say dark treatment. So, as shown in this top figure, the COG7 mutant, after 3 days of dark treatment, has comparable levels of yellowing, which is a visual indicator of plant senescence, the onset of it, to the wild-type plants after 9 days of dark treatment. Now, these dark conditions are also sugar limiting, right? Because plants need light to perform photosynthesis and synthesize sugars. But of course, in dark conditions, they don't have that light. Interestingly and counterintuitively, we see an increase in protein glycosylation, or the attachment and modification of carbohydrate groups on proteins at the onset of dark-induced senescence, despite sugar being limiting in these conditions. So this bottom figure, which is an immunoblot, it uses an antibody to detect a particular sugar, in this case xylose, that is part of the carbohydrate groups on some glycosylated proteins. What we can see is that in this COG7 mutant, right, which senesces, has an earlier onset of senescence compared to wild-type plants, when there is no sucrose supplementation and after 3 days of dark treatment, you can see, as indicated by the darkness of the band, there are comparable levels of glycosylated proteins from a specific glycosylation pathway to wild-type plants with no sucrose supplementation and after 9 days of dark treatment. Right? This is in line with that earlier onset of dark-induced senescence that we see in the COG7 mutant. Now, of course, there are many glycosyl transferase enzymes, or enzymes that actually perform these glycosylation reactions within Arabidopsis. And some, as in this bottom figure, have been studied in the context of dark-induced senescence. Others have less so. And in fact, there are two glycosyl transferases called GT1 and GT2 that show increased transcript levels at the onset of dark-induced senescence, which would indicate that there's a chance that they're playing some sort of a role in this onset of dark-induced senescence. So my research is actually investigating whether these two genes, GT1 and GT2, are involved in the onset of dark-induced senescence. And so, in other words, I'm looking at the function of these two genes. And within this overall work that the lab is doing, there are going to be two main approaches that will be taken to look at their function, using knockout mutants and overexpression lines. My work is going to focus on using these knockout mutants, or plants that have non-functional versions of the GT1 and GT2 genes. So to generate these knockout mutants, CRISPR gene editing was used. Using the flower dipping method of plant transformation, the gene editing machinery for both GT1 and GT2 were transformed into Arabidopsis, ultimately allowing us to generate transgenic seeds. Now, not all of the seeds are actually going to be transformed, so we needed to select for the successful transformants, or seeds that actually have the gene editing machinery, using an antibiotic resistance marker gene. Now, those two steps were performed prior to my time in the lab, but where I picked up is with this third major step, genotyping. So, even though all these seeds at this point that we're using have the gene editing machinery, it's not guaranteed that they'll actually have a gene edit, or even a gene edit that would likely result in a loss of function in the two genes. And so this genotyping step is broken down into two parts. The first is to check whether or not there is some sort of an edit in the GT1 and GT2 genes. And that has been done using restriction enzyme digestion and gel electrophoresis, which I will talk about more on the next slide. And then in those plants that actually have a mutation, we want to see what is the specific mutation. So what is the sequence change to the genes that has actually occurred. And then from that, we can see whether or not the genes will have likely lost their function. And finally, using those plants, which are our knockout mutants, we will then put them under dark treatment and then compare their onset of senescence to that of the wild type. And then from that comparison, we can infer whether or not these two genes are involved in the onset of dark-induced senescence. So the way that this restriction enzyme genotyping system works, is that in the wild type allele, we have this restriction enzyme digestion site. And once we treat a PCR amplified fragment of the wild type alleles with a restriction enzyme, that restriction enzyme will cut the DNA, producing two smaller fragments from one larger one. In the mutant alleles, however, we don't have this restriction enzyme digest site. So when we treat it with them with restriction enzymes, there's going to be no digest, right? These fragments are going to stay the same size, and they'll actually be bigger than the digested wild type fragments. And so using DNA gel electrophoresis, we can actually differentiate the mutant alleles shown here in these blue boxes from the wild type alleles that are unboxed based on the size of the fragments. And so overall, what we're seeing is that we have multiple mutant lines, and as indicated by these kind of more slight size variations in the fragments, right, these slight differences in the band positions on the gel, we have at least a few independent, or in other words, unique mutations among these mutant lines. And this is exactly what we need for our final step, which is to actually take these mutant plants and then put them through a phenotype assay, right? Put them under that dark treatment and observe the onset of their senescence. And so based on this prior work that had been done in the COG7 paper that I had talked about on the introduction slide, I expect that knocking out these genes, GT1 and GT2, will cause mutant plants to have a delayed onset or slower progression of dark-induced senescence, as depicted here in this graph. But of course, we can't really say that until we've tested them, so depending on exactly what their function is, right, you could even have accelerated senescence or no difference. But regardless of the result, this work will contribute to our understanding of the machinery involved in the onset of dark-induced senescence and our overall understanding of plant stress responses. And with that, I'd like to thank my PI, Katie Dehesh, as well as my mentor, Ginger, and the NSF for funding both my research and the Plant Bio REU program. Thank you.